Hello to everyone watching. My name is Shayla Perez.
Litzy Mendoza.
I'm Neat Dylan.
And this summer, we all had the amazing opportunity to participate in Dr. Stokes' lab thanks to the SURE program here at CSUB. In our lab, we got to work with TTX, which is short for tetrodotoxin. It is a small molecule that is found in a large number of organisms like pufferfish, mollusks, and bacteria. Another thing we also got to work with was Taricha granulosa, which is a type of salamander referred to as the rough-skinned newt. This newt also has TTX. So TTX is a neurotoxin that stops signals moving through nerves, causing paralysis. So if ingested, it can lead to death due to asphyxiation. So therefore, the newts use this toxin to defend themselves from predators. So TTX in the newts is highly concentrated in their skin and also in the ovaries of female newts. But we still don't fully understand where TTX in the newts come from. One prediction is that it is produced by the newt, the other is that they get it from their diets. So the first part of our research was investigating whether the metabolic rates in newts increases during their TTX production. We presume that if a newt produces TTX, it is likely that it takes a lot of energy. So both the newt's metabolic rates and TTX levels were measured. To understand this better, the metabolic rates of newts were measured before and after secretion of TTX from their skin glands. Secretion of TTX is done by applying low voltage electrical stimulation. This causes the TTX levels to decrease. They then build those levels back up. This process is done by the newt, likely costing energy, so their metabolic rates would increase. We have been working on quantifying the TTX levels from the newts before stimulation as well as 11 and 30 days post-stimulation. The first step is to extract TTX from the newt's skin. The skin punches were then grounded in a tube of acetic acid using a pestle, as seen on the picture on the right. The samples are then boiled to eliminate proteins, and lastly, the samples were centrifuged and filtered to purify them, as seen in the photos on step 2 and step 3. We are then able to quantify the TTX in each sample using a competitive inhibition enzymatic immunoassay.
The following day, we quantified TTX. We first pipetted conjugate into a microwave plate. The conjugate has TTX bound to the bovine serum albumin, known as BSA. This will bind to the bottom of the plate. In between each step, we wash the plate so that we are left with only what is bound to the plate. We then add BSA, which fills in any gaps so that we don't get TTX binding where it shouldn't. We then add standard samples and primary antibodies to the plate. The antibodies will bind to the TTX in the standard sample or to TTX in the conjugate on the plate. So samples with a lot of TTX will have very little primary antibodies bound to the plate. After this step, secondary antibodies are added. These bind to the primary antibodies. Lastly, PMPP is added. This will bind to the secondary antibodies and change colors. We then measure the absorbance of how dark the color is using a plate absorbance reader. This allows us to quantify the amount of TTX in the sample. On this slide, you can see my colleagues and I completing each step, from pipetting conjugate, BSA, preparing the secondary and primary antibodies, the PMPP, and placing the plate in the plate reader. The last two images are shown steps that have been done in between each of the steps, such as the incubation for an hour and washing the plate. Once complete, the plate looks like this. And the wells that are darker in colors have less TTX. Wells that are lighter in color have more TTX. We still have more data to collect. However, this is what we have so far. We expect that the 11 and 30-day samples should have less TTX than the before sample. We see this in most. For example, sample number 3534 has a higher TTX before the electrical stimulation than after. Again, some still need to be further diluted to get accurate numbers. These data will be correlated with metabolic rates. Stay tuned.
In addition to measuring TTX levels from newts, we looked at whether newts glow, or technically biofluoresce. It has been known for a long time that many fish are biofluorescent, as are many other types of animals. Biofluorescence may have a lot of different functions. It may be used to communicate to other individuals or as a warning to predators. Recently, it was found that many amphibians are also biofluorescent. However, Taricha were not tested in this previous work. We also tested whether newts also display biofluorescence. So we set up a camera with a filter and a blue excitation light attached to the tripod. We placed the newt and the camera in a portable photo booth to keep out the light. Filters allow human eyes to see the fluorescence. We don't yet know if newts or their predators would be able to see any of the fluorescence. But we found out that newts do biofluoresce. The photo on the left shows the newt under the blue excitation light with no filter. The images on the right show the newts with the filter. You can see that some of the areas of the newt do fluoresce. We need to do more work to identify any patterns, but as of right now, males seem to fluoresce more than females. We still have a lot more work to do for both of these projects. We'd like to thank Dr. Stokes,
Bela Sundan, Danica Bergen,
Andrea Medina,
the Title 5B Graduate Excellence Program,
and Chevron for this wonderful experience.
and a special thanks to Dr. Doty and Jonathan James for their work on the metabolism project. And here's our literature cited, and pictures of us throughout the past 3 weeks of doing newt care and much more. Thank you.
Lead to death due to exfoliation. The newts are
asphyxiation.
hurting us.
So let's redo that.
Grounded in a tube in acetic acid. Wait, what? I said that totally wrong.
To the bovine serum albumin.
Fluorescent. However, Taricha were not tested in the also display and no.
Danica Bergen.
Andrea